Thank you, Federico, for introducing me, in case you have actually done so, because since this is a recorded presentation, I have no idea. Well, as Federico might have or might have not said, my name is Alicia Mastretta. I work at Conavio as a CONACYT Research Fellow, and today I'm gonna present you a talk that is more, let's say, food for thought than actual data. What I want you to wonder in this talk is, what are we doing in bioinformatics, and how does what we do came to be, and what's next? Where are we going? And importantly too, how do we build what is next? So let's start by answering, what do we study with bioinformatics? Which, by the way, is something you should really know. And if not, you are in the wrong conference. Okay, so well, you will say, yeah, I am in the right conference. I know we study DNA sequences in bioinformatics, and some of you will say, and especially, very importantly, also proteins. And this is right. You will also say that this study is done by developing different softwares or making use of the data that is already developed. And you are right, this is bioinformatics. Some other of you will say, well, this is very important for biomedical research. We're studying, for instance, cancers or diabetes in human beings. And others will say, and also in human beings, we are studying our own diversity as a species. But of course, there are others who apply these bioinformatic tools to answer questions in other species, especially with an evolutionary focus. So this is correct. All of this is bioinformatics. Welcome to the right conference. But just think about it. Biodiversity is more than only the genetic diversity that is within a species or closely related species. It also encompasses different species to different ecosystems and the very complex interactions that are relating all this biodiversity. So if we know that biodiversity is all of this, why is it that bioinformatics kind of like stays from the molecular processes that happen within cells to the genetic diversity within a species and somehow also in between other different species? But when we came to questions of taxonomy and biogeography or ecology, then it comes biodiversity informatics to answer these kinds of questions. For instance, to address where species are distributed and why. But when we talk more broadly about ecosystems, landscapes, and environmental science, then some even separate a new realm, which is ecoinformatics, as a different field. So therefore, even if biodiversity is all this wide range of life on Earth and very complex processes that we know are entangled from the genetic to the ecosystem level, we restrict each of them to a particular subfield of informatics applied to biology. So why are we doing that? Why are we studying biodiversity in these separate tools? Well, I would like to start with a bit of history. So how does that came to be? You will say, let's start with the history of bioinformatics. Some of you will say that the history of bioinformatics is closely related to the history of GenBank, therefore starting in 1982 when it was the first GenBank release. Some other of you will say, well, yeah, and after the Genome Human Project and the release of the whole genome sequences in GenBank, then it's when it started the boom of really applying informatics and all sorts of new computational methods to big data. But that's not true. That's not when bioinformatics started. Bioinformatics, to start with, didn't start with DNA sequences. It started with protein sequencing in the 50s. And the first ever bioinformatics software, and therefore the first bioinformaticians, was released in 1962 by Margaret Dayhoff and Robert Ledday. Margaret was an American physical chemist, and she was the inventor of bioinformatics, we could say, because she pioneered the application of computational methods to biochemistry. And then Robert, he was a physicist, and he also sought to combine computational tools to biomedical problems, and together they developed ComProtein, the first ever bioinformatic software, which was used to assemble protein sequences. If you wonder how that looked, on the left you can see the computer that was used for it, and of course this program was written in Fortran, and you need all these cards to run it, and I have no slightly idea of how this was done. So please don't make me run that program. Okay, so then we continue with protein sequences with the first multiple sequence alignment, which was done in the 70s. And it was not until the late 70s with Sanger sequencing when we can start talking about using DNA sequences for bioinformatic analysis. Very importantly, as this started to be developed, from the beginning it was sought to incorporate all these different data into proper databases held by an institution. This is when the European Molecular Biology Laboratory built the first data library. And then we finally get into the plot below. In 1986 and 87, EMBML, Gene Bank, and the DNA Data Bank of Japan, they joined their efforts to have a united database, particularly to standardize the format and define the minimal information that was needed for reporting. Okay, and then very importantly, in the 90s is when the internet was invented and of course was applied to this new research. The EMBLM was the first sequence library to be available on the internet, followed by GenBank. And the NCBI site was also made available in 1994. So it was, before was the internet, and then we have the whole genome, the Human Whole Genome Project, and then all the whole genome sequencing that came afterwards, and that is when really things exploded into big data. Okay, so I want to highlight this quote from NCBI site because interestingly, NCBI was originally created to basically make databases and the computational tools that were needed to make it available. However, through the widespread use of the internet, NCBI became more inclusive aware of the role of pure biological research, that is, to develop bioinformatics. This is also from the website of NCBI, and this is what they realized they were doing after the 90s. I'm going to take a sip of water while you read these quotes, which I think they're cool. Basically, nice mug, isn't it? I know. Basically, we're talking about developing new methods to deal with big data, to create computerized systems for storing that data and applying it to molecular biology studies, just making the data available, providing access. So that was the history of bioinformatics in a nutshell. And now let's go to the biodiversity side. Well, this started quite earlier than that because it started before the Western science was developed. In the case of Latin America, it started in the codexes that different Mesoamerican peoples used to record the diversity of species, especially plants and animals that were present and the uses they had. It was then after Linnaeus published the Systema Naturae that the Western science was born, and new scientific collections like herbariums and museums started to be done to record biodiversity. And then, of course, this was done at different speeds depending on the type of organisms. Okay, but importantly, none of this was in computers. Okay, all of this was recorded in notebooks, in museums, in some sort of, even in the case of herbarium, it was in the list of specimens itself next to the pressed plant. So in the 90s, when computers started to be available to scientists worldwide, then the question arises, well, shouldn't we digitalize biological collections? And you and I will think that, yeah, well, that's the thing to do, right? Even supermarkets are making their things digital. Why not? So what I want to share here with these quotes that actual people said is that there was a lot of reluctance to do that. So first of all, people thought that it was going to be computationally impossible just to go and put all the amount of people you will need to translate from the paper to a computer the data. Others thought that not even the computers were ready to handle this kind of disperse on different databases and to exchange the data as it will be necessarily. But also there were more, let's say, social concerns of people that they were reluctant to even share the data, as you can see in this quote and in this other. As we know today, that's a thing of the past, because today the Global Biodiversity Information Facility has free and open access to biodiversity data, including billions of occurrence records of species worldwide, and gathering information from more than 1,600 institutions. So this is as of yesterday yesterday, which is 2 weeks before or 1 week before you are watching this. So what happened? I'm going to tell you what happened to make this possible, because even if it's not very related to bioinformatics, it's very related to the history of science in Latin America, and we played an important role. So I think it's important that we present this thing in this conference. So let's put ourselves at the early 90s, when the Convention of Biological Diversity was signed by different countries worldwide, especially by mega diverse countries like our countries in Latin America, but also others. Some countries were very keen on developing very early in the process national, let's call them, biodiversity agencies. And these countries were Australia, Costa Rica, Mexico, with Conavio, where I work, and Indonesia. And these were the firsts. Australia had developed in their agency not only an institution that would put all together the records of where their biodiversity was, but also they developed a software for ecological niche modeling. That is, to use the occurrence data of where a species has been found with environmental layers to then produce a model of where in Australia, in this case, the species could be found. Now, to learn about this, country representative scientists from Costa Rica, Mexico, and Indonesia went to visit the houses to learn about this thing. Mexico immediately was very interested into this and decided to apply it in our own country. However, there was a problem. Data on Mexican biodiversity was dispersed. A good part of it was at UNAM in the Mexu herbarium for the case of plants, for instance, but most of it also was in foreign herbariums in different countries, like this one in Texas, but also different institutions in Mexico that didn't talk to each other, like Durango, who belongs to CD Politécnicos in the staff, and also other institutions, for instance, in Yucatan. So what Mexico did, very interesting, is that they pushed for having a project called the North American Biodiversity Information Network, signed between Canada, Mexico, and United States, so that Mexico could get the data of the specimens that were collected in the Mexican territory, and to get that data and also to share the data together in a single database that Canada, the US, and Mexico could see. And they did this, and it was very successful. They managed to do what, some previous slides before, people say that it was going to be impossible. So Mexican scientists with colleagues from Canada and the US showed that it was possible to do it. And then this was so successful that after a 1999 meeting of the OECD Mega Science Forum, Mexico and other countries proposed that a global biodiversity information should be needed. So therefore, they proposed to have the creation of GBIF, which is a very successful project that I just showed you. And something I want to highlight here is that it was not only about getting the data, but sharing the data and connecting it using the internet. So today, Conavio keeps the data of different Mexican institutions and makes it available to the public, and the data keeps really growing, as you can see in this figure. However, it was not only having the data for the sake of it, but also new applications came with it. For instance, if you remember the niche models I mentioned earlier, Conavio decided to do that not to predict where a species is, but also to predict where a species could be. And this became a very important question for invasive species, and therefore for decisions that the government could make regarding, for instance, whether to introduce or not a particular bee for pollination. Okay. So if you think about it, what occurred was some sort of parallel evolution between bioinformatics and biodiversity informatics. And I didn't talk about ecoinformatics for the time, but it's a very similar story, where first different institutions had to agree to interconnect databases and to have standards. Also, all of them agreed to put the data available online using the newly born internet. And thanks to that, the amount of data that was gathered was far larger than what could have been possible if a single institution were working with. And so what? Well, making that amount of data allowed to develop new methods to deal with a lot of data, but also to generate new theories and new applications that emerge from having the data available. So, as I said, if you put a bike lane, then you harvest cyclists. Similarly, if you put the data available, then you harvest new theories, new applications, and even new fields of knowledge. So what's next? Is that enough, what we have been doing? What's the next biggest step? Should we just keep accumulating data, or is there something else to do? So as I told you before, biodiversity encompasses from the genetic diversity to the ecosystem diversity and all the interactions within. Currently, this is separated, and we really talk to the people doing ecological studies or evolutionary studies at higher levels. However, I think it is now time to start thinking on integration. That is, to consider questions that can occupy data at all these levels, but also that require new tools to make possible to answer those questions. Of course, I am not the first one to think about it. If you go to this very nice paper by Peterson et al., they actually propose how to interconnect the different data sources, and also to think about the type of questions that could be addressed if we did that. So what is needed to get there? In summary, there's 3 important points. First, we need to deal with data quality, because a lot of big data, you know, is just big shit, and your algorithms are as good as the data you feed them with. Also, we need to deal with problems of scale, because it is not the same algorithms to answer questions at the molecular level or a species level or the ecosystem level don't need the same quality of geographic data, for instance. Secondly, we need to integrate data that it's very different to each other and that lives in disparate databases. And most importantly, the last of them, we need new conceptual frameworks and new questions. I think the first key landmark was to get all this massive data together. And that's a thing of the past. Doesn't mean we shouldn't keep doing it. It's just that the next landmark, it's going to be to have new conceptual frameworks and new questions, not just to keep accumulating data and see what happens. So we need to go beyond the bike lanes and now have the entire city or something like that. I'm going to show you some example questions that I have been thinking about for a while, and that will need to have these bridges across different levels and different informations on biodiversity. So first, how does genetic diversity changes in space and time given land use and human activities? Some sort of genetic diversity monitoring, similar to what we do with species. Also, can we predict the future distribution of alleles? And if so, could we speed it up so that the species could better adapt to climate change? How does community assembly, and I'm thinking from bacteria to plants altogether with fungi and even animals, influences expression patterns and phenotypes? How much our phenotype is due not only to the interaction with the environment, but also with the other species that form part of the environment? And finally, does ecosystem degradation increases pathogens availability? For instance, this is a question that I started to think now that we are in the middle of a pandemic. So that's all very nice, but the paper I show you of Peterson and colleagues was from 10 years ago. So what's going on? Why haven't we done it? How do we really build what is next? So I think we should just, do it and let the others follow. So I'm going to show you what we are doing at Conavio very briefly. So regarding data quality, we are moving from people recording data in lab books or notebooks in the field, and then copying that data to Excel, putting it in a hard drive, so that eventually after the project, when we are about to publish it, it will perhaps get to a data warehouse. Instead, what we are doing is that in our projects, we are using this tool called Kobo Toolbox. It's a free software developed by Harvard and others, which allow to collect data using forms similar to Google Forms, but something that you can fill on the web, but also in your smartphone in offline conditions. And importantly, this allows us to control humans' errors, especially by not allowing humans to write things up. For instance, you will have to select from a closed vocabulary. And this is very important because you will be amazed of how many different ways people can find to write maize. It's just unbelievable. Okay, so then researchers produce this data, they give it to Conavio as they are collecting it, because Conavio is Kobo is installed in our servers, so we continuously have access to the data, even if a project is just ongoing. In Conavio, in certain given times agreed with the researchers, we perform a data quality evaluation. If there is something that doesn't seem right, then we tell the project researchers and they correct it and give it to us again. So after the project is finished, then we can finally integrate this data in Conavio databases and make it available to the public or wider researchers. Now I want to talk about data integration considering disparate databases, which is one of the key problems we have to tackle to move forward, remember? So I want to talk about a project we have on Conavio on agrobiodiversity, that is diversity related to how we produce our food, from the genetic level also to the interactions and the different species that we don't eat but that form part of the agroecosystem. So we have all sorts of disparate, very different data here, from occurrence records of where things are geographically, but also we have phenotypes associated to those records or performed independently as part of an experiment. We also have a lot of information from, let's say, from social sciences that are qualitative, not necessarily quantitative, and are very interesting and difficult to put into a database. Also, we have nutritional information that comes from years of other people measuring the nutrition quantities of different foods. And of course, we also have species diversity, genetic diversity, and all that biological stuff that we normally have already. Okay, so putting together all these disparate databases is not a trivial task. So the last 2 years, we have been developing a new software called Centro. You can see there our GitHub documentation and also our GitHub projects. And this is something we have been doing in Conavio in collaboration with our German partners from Jülich. So Centro is a software tool that allows us to quickly create a data warehouse tailored to the specific specifications of a project. How it works is that you tell Centro what the structure of the data is, that is, you define your data models and where the data will be stored, and then Centro automatically builds two interfaces, one graphical user interface, which is very intuitive, and an API which allows to perform more sophisticated queries. In both interfaces, you can create, read, update, and delete your records as you will normally do with a database. We also have integrated custom validation filters and a system to integrate plots. And most importantly, our system allows to have data in different servers that might even have different storage technologies. And we did that by using GraphQL, which is a technology developed by Facebook, which allows to integrate data from different data storages in a single database that responds to a query as if the data were in a single place, although in reality it may be in different instances. Okay, let me give you an example. Let's imagine that we have a scientist here who is measuring sugar content and number of fruits of tomato varieties. And then she will need to develop the data models of this experiment, which will be the tomato plant and the number of fruits and the sugar content. But also, let's imagine that she also has genetic data in a different database that was generated by other colleagues. What she needs to do is to establish these two data models, and then something happened to the animation. And then feed Centro with it, and Centro will create the two interfaces that I mentioned earlier. The graphical interface looks like this. You can click on the plain variant, for instance, and then you will see here on the right a small view of it. And you can also upload, delete the data here. I will, in other occasion, show you a live demonstration of how it looks. The API looks like this. You can write very sophisticated queries using GraphQL. And of course, you can use this query to get the data from your favorite programming language like R or Python or whatever. So we're applying Centro right now to develop agrobiodiversity information system where data of these all different sources and others I didn't talk about will be stored and interconnected. We hope that with this, we will be able to answer questions like the following and among others. So what are the distributions of domesticated plants, their wild relatives and varieties of these plants? How do management, production practices, and interactions with wild relatives affect the genomic composition of domesticated species? Is agrobiodiversity positively associated with the food security of the people that are safeguarding it? And finally, can seed exchange be assisted to provide campesinos with the variation needed to cope with environmental change? So these are just some few examples that we couldn't address if the data were all dispersed. Thank you very much. I hope that this talk was useful to let you thinking on what are the new challenges for bioinformatics and how Latin America can contribute to solve them. I also would like to acknowledge our funded sources from the Mexican side and our colleagues in Germany. Thank you very much for your attention, and I am happy to take questions.